chapter, and he ran the raffle for about 20 years. He was a great fly fisherman, a wonderful teacher, and just a tremendous fellow. And we were delighted to name the uh, scholarship fund in his name. This year, as every year, we review applications for a recipient for the scholarship. And we got about a half a dozen applications. Allie and I reviewed those, and it's really incredible the resumes of these young people. Um, when I look at what I was doing in college and what these folks have done, it's really amazing. Um, Jordan Trujillo was raised in the San Luis Valley. She went to Western Colorado University where she graduated magna cum laude. Lily at CSU, she's been a graduate student in fish, wildlife, and conservation biology. And she really has done a remarkable number of things. She's been a graduate teaching assistant, a research assistant. She's worked for Colorado Department of Natural Resources and a couple of the ranches she's going to talk about in the San Luis Valley. Um, spent some time at the University of Idaho on just nematode research. That's fascinating. I have no idea what that is. The city of Gunnison for their natural quality water laboratories, and I mentioned for the Colorado Parks and Wildlife. And so it's my pleasure to introduce Jordan, who's going to be talking about stream health and restoration in the Rio Grande Valley for the trout. Give a warm welcome for Jordan.
All right, hello everybody. My name is Jordan, and thanks for having me here today. Um, is it on? I just wasn't talking close enough. But I just want to start off with an outline of my presentation today. First, we'll discuss Trinchera Ranch, which is where my research took place, and then we'll get into bug stuff, which is what my master's thesis is based on. So these slides will be a little bit lengthier because we have hypothesis and methods and results associated with them. And then into our fish stuff, which is where we'll discuss whirling disease and how it's managed on Trinchera Ranch. So within I just turned it off. Okay, are we good? Okay, within Colorado, there are 6 major drainage basins flowing either east or west of the Continental Divide. In the case of the Rio Grande flowing south. The upper Rio Grande drainage basin encompasses about 8,000 square miles of Colorado, with about a third of that area held within a closed basin. Meaning there are no surface water outlets contributing to this Rio Grande River. Water within this closed basin is held in underground aquifers are diverted and stored to San Luis Lake, located just south of the Great Sand Dunes. My study area is situated in Costilla County, surrounded by the Sangre de Cristo Mountains, just outside this closed basin. So headwater systems of this area do contribute surface water to the Rio Grande River. However, some segments of these watersheds have been significantly altered from their natural flow, with some portions running completely dry in order to meet water demands. My study sites are located on Trinchera Ranch. This ranch is highly devoted to conservation, with most of the property being held under conservation easements. The ranch has resident populations of Rio Grande Cutthroat trout, a species that was recently up for listing as endangered. These fish are found above natural waterfall barriers in the upper segments of some rivers, where brook trout have yet to outcompete them. The expansiveness of the ranch allows us to study streams through elevations and various habitat types. And because Trinchera Ranch is managed as a working ranch, We can study the effects of different human activities like agriculture and timber harvest on these streams. We'll start off with my bug stuff, which as anglers, I hope you all appreciate as much as I do. And I'll start by introducing my research questions, which are how do bug communities change through different stream orders, and what are the roles of environmental and dispersal-related mechanisms on shaping these communities? In order to answer question one of how bug communities change through stream orders, we need to define how we're going to measure this change. And a common way to do that is through alpha diversities. Alpha diversity is just a fancy way of saying what bugs can we find at this site. It's the local site diversity. To calculate alpha, you count the number of bugs found at a site. Site A has a diversity of 4 because of its 4 distinct bugs. It has a snail, a mayfly, a caddisfly, and a beetle. Whereas Site C has an alpha diversity of 2 because there are only 2 unique bugs within this site. It has a singular caddisfly and a group of mayflies. And because all of these mayflies are the same species, there's only 4 unique bugs found at this site, with the alpha diversity being 2. So we want to understand how these diversities change throughout the stream gradient. We predict that as stream order increases, bug diversities will increase. So as you move down the gradient, communities from larger streams will have more bugs and larger diversities. Based on this hypothesis, we expect Site D to have the largest alpha diversity because of its location further down the stream. This is the idea that as streams become larger, they can support a greater number of bugs, leading to larger and more diverse communities. Whereas these smaller headwater sites are more isolated, limiting the number of bugs that can live there, decreasing their diversities. For question 2, we want to understand what shapes these communities, and specifically looked at how dispersal and environmental factors work on shaping these bug communities. For bugs, dispersal is a means of travel. Whether it's flying from stream to stream like a mayfly or swimming through creeks like a scud. Dispersal barriers can shape bug communities by stopping travel and preventing bugs from occupying certain areas. These can be overland distances like mountain ranges, acting as barriers between streams, or in-stream barriers like waterfalls, preventing the upstream travel of some insects. And although these are barriers, do not consider them bad or harmful objects. They are simply working to shape communities. These waterfall barriers are shaping fish communities by preventing the upstream dispersal of non-native fish into native fish habitats. One way we are testing the role of dispersal is through distance relationships. This is the idea that greater distances between sites will increase differences in communities. So the further away sites are from one another, the less likely they are to be similar. Based on this idea, sites B and C are relatively close, so they are likely to have similar communities, where sites B and C are far away from one another, so they are likely to have different communities. Local environmental variables, like temperature and topography, anthropogenic features, or any factors that shape a habitat, can also shape bug communities. To test the role of environment, we looked at the habitats themselves and considered the local variables contributing to and shaping those habitats. We predict that similar environments will have similar bug communities. So sites B and D will have similar communities, while sites C and D will have different communities. This will inform us on the role of environment in shaping bug communities. In conclusion, we predict an increase in distance and a difference of environmental variables will lead to differences in community composition. So as sites become further away, or as environmental variables are more different, communities will also be different. We looked at 3 watersheds for our bug study: Sangre de Cristo and Trinchera. These watersheds all drain into Smith Reservoir before continuing on to the Rio Grande. This map does a really good job at showing the discontinuities within this system. As you can see, this lone segment coming off of Smith Reservoir. This is the old confluence with Trinchera Creek. But now you can see that Trinchera Creek is diverted north just after Mountain Home Reservoir in order to meet water demands within this system. Within the ranch, we collected data from 63 sites representing 23 first-order streams, 18 second-order streams, and 22 third-order streams. These sites were collected from an elevation gradient of 7,900 to 12,000 feet. From our 63 sites, we established a sampling reach. Within the reach, we subdivided them into 6 cross sections, and wetted width, depth, slope, canopy cover, and 5 pebble measurements were taken from each site. The presence or absence of stream diversions was also collected at the site, and grazing intensity was a visual estimate measured along the reach. Elevation was recorded at the macroinvertebrate sampling site, where a 1-minute pigment sample was taken from a half a square meter area. Stream order was recorded in truth in the field and truth looking at satellite imagery. Key environmental variables were calculated using GIS, the upstream watershed area, and the anthropogenic land cover, which was calculated from a 100-meter buffer around the site and included measurements of ag, timber harvest, railroads, and ranch roads. From each site, the macroinvertebrate kickneck samples were sorted entirely. So every single bug from every single sample was counted, ID'd, and 10 length measurements were recorded per site per family. I also want to introduce EPT, which are these top row of bugs up here. So Ephemeroptera, Plecoptera, and Trichoptera. So are mayflies, stoneflies, and caddisflies. These bugs are commonly used as indicator species because of their sensitivity to stream conditions. So the presence or absence of these guys would indicate some implications of stream health. This pie chart represents all of our bugs sampled. It's broken up by taxonomic order, and the slices are their abundances across all of our 63 sites. For our sampling efforts, we ID'd 18 orders represented by the different colors. Within those orders, we ID'd 60 unique families. And as you can see, Diptera and Ephemeroptera, so our midges and mayflies, made up about 2/3 of our bug communities. So just putting some perspective on the bugs that make up our communities throughout our sampling system. So to answer question 1 of how bug communities change through stream orders, we looked at how alpha diversity changed with stream order. So of our 63 sites, did diversity change across these first, second, and third order streams? We used 2 data sets for this assessment. This first plot, the top plot, includes all of our bugs sampled, and then the bottom plot is our EPT bugs, so our mayflies, stoneflies, and caddisflies. For our analysis, we found that diversity did not change with stream order. This means the number of bugs remains consistent throughout our sites. So when looking at these plots makes sense, because for all our bugs, for first, second, and third order sites, it remains about 23. And for the EPT bugs, it's about 12 for each of our sites. So these numbers remain consistent, suggesting diversities didn't change with stream order. Originally, we predicted diversity would change with stream order, where diversity would increase as stream order increased. However, our results found diversity did not change, suggesting that each stream order supports a similar number of bugs. This goes against our hypothesis, but does suggest some interesting things about bug communities. Which brings us to our next question. What are the roles of environment and dispersal in shaping these bug communities? We tested this by testing dispersal distances between sites to inform on the role of dispersal. So our bug community is different due to things like mountains and waterfalls preventing dispersal and stopping certain bugs from inhabiting certain sites. We then compared our environmental variables across sites to see if differences in environment led to differences in bug communities. Originally, we predicted an increase in distance and differences between environment would lead to differences in communities. We found support for this with some of our tests. We found distance had no role in shaping bug communities, represented by the flat line from our distance factor. This suggests bugs are not limited by dispersal. Though even sites that are miles and miles apart from each other had similar bug communities. Environment did have a hand in shaping communities. So sites that were environmentally different from one another also had different communities, represented by this positive upward direction of this chart. In conclusion, for question 1, we found diversities were similar across streams. But for question 2, we found environment shaped different communities. So how does this work? How are diversities the same, but communities different? These results suggest there's some sort of species turnover happening across these stream gradients, which is the idea that as you move down the stream, one bug moves out and another replacing it. So there's this constant replacement happening across stream orders, keeping diversity similar, but communities different. And the reason communities are changing is due to differences in environment. In this example, all of these sites have a diversity of 4, but they are made up of different bugs, representing different communities. And these communities are structured due to environment. So this third order site has a crawdad in its system, which are usually associated with slower moving warmer waters, which is a different environment than the first order sites, which are cold and fast flowing. These environmental differences are what cause differences in bug communities. So what does all of this bug stuff mean? So even though diversities remain the same, we found that first, second, and third order sites supported distinct communities. These communities are influenced by environmental factors, resulting in different assemblages at each stream order. Together, these stream order communities play a critical role in shaping whole watershed biodiversity. So recognizing there is a spread of bugs throughout the stream system emphasizes the importance of entire network conservation. And knowing how communities naturally vary across the stream gradient is important for effective management. Understanding this natural variation between bug communities prevents misinterpretation of community differences as degradation. This leads to more informed management decisions, improving restoration efforts and conservation priorities, especially when deciding where concentrating efforts for fish management. Which brings us to our fish stuff. And since Trinchera Ranch is located within the Rio Grande drainage basin, our fish of interest are, of course, the Rio Grande Cutthroat trout. Like our bugs, we looked at 3 watersheds, Sangre de Cristo and Trinchera. Within the ranch, there's still some resident populations of Rio Grandes. These fish can be found above natural waterfall barriers, like on Little Ute Creek within the Ute watershed, on Squirrel Creek within the Trinchera watershed, and in the upper reaches of some streams, where brook trout have yet to outcompete these natives. Trinchera Ranch has completed multiple reclamation projects to restore these fish back to their native range by implementing impassable fish barriers and removing non-native brook trout to reintroduce these Rio Grandes back to their native habitat. Complete restoration and reintroduction have been done within this Placer Creek drainage, a tributary to the Sangre de Cristo. This restoration project was extra special because not only did they reintroduce Rio Grande cutthroat trout, but they also eliminated whirling disease from the system. They did this by breaking the life cycle of Myxobolus seborrheilis, which is the causative agent of whirling disease. As you all probably know, whirling disease can be detrimental to fish populations. If fish are infected severely enough, it can cause deformities and even death. There is currently no known cure for whirling disease aside from breaking the life cycle. Which is a pretty complex life cycle. It requires two hosts, a worm host and a fish host, specifically trout. And rainbows and cutthroat trout are highly susceptible to infection. So I want to start off by getting into the whirling disease cycle so we understand its complexities. Whirling disease spores are found within streambed sediments. These spores are ingested by the worm hosts, which are then transformed into a new spore form called a TAM. TAMs are released from the worm host and into the water column, where they free float until they find a fish host. These TAMs enter and infect the fish through the skin and gills. And once in the fish, these TAMs migrate to the head and spinal cartilage and begin reproducing. This step can cause clinical stages of the disease to become present, so blacktail and whirling behavior. Within the fish, these TAMs convert back to spores, and then these mature spores are released from the fish and settle back into the stream sediment, starting the cycle over again. By eliminating one of these hosts, we can break the whirling disease cycle, and a common host to eliminate is the fish, which is what made the Placer Creek reclamation such a success. By taking out the fish host, we broke that life cycle, eliminating whirling disease from that system. Trinchera Ranch was interested in testing for whirling disease in relation to their reclamation projects. In the summer of 2022, when I first came to Trinchera, the North Fork Reclamation Project was already in place, with plans to complete the South Fork Trinchera project in following years. So I focused my efforts on collecting samples from within this Trinchera watershed to inform on the presence of whirling disease within these reclamation sites. If whirling disease is present within these locations, it would halt the reintroduction of Rio Grande Cutthroat trout in order for that whirling disease life cycle to be broken. So our goal was to test for the presence or absence of whirling disease within those reclamation sites. And because whirling disease has such a complex life cycle, we focused our sampling efforts on three main areas: collecting the worms themselves and testing them for whirling disease, collecting fish and processing them for spore counts, and collecting water samples and testing for environmental traces of DNA. I started off by collecting worms throughout the Trinchera watershed. The worm hosts are T-tube effects worms, or commonly referred to as sludge worms. These worms inhabit muddy sediments of slow-moving warm waters and are typically found in large colonies. My efforts involved sampling 108 sites within this Trinchera Creek drainage. To collect these worms, I walk downstream looking for suitable worm habitat. Once I find a location, I take 1 to 3 scoops of sediment per site and then rinse the sediment in the water to look for worm hosts. To look for the worm hosts, you just root around in the mud, which sounds a little ridiculous, but because these worms are typically found in large colonies like this, they're easy to find. But unfortunately for me, none of my samples, of my 108 samples, were this large. But when I did find worms, I would pick out 50 individuals and test those for DNA. Of my 108 sites, I only found worms in these 3 locations. And these worms tested negative for whirling disease. Since my worm efforts proved futile, we decided to also collect fish from the Trinchera Creek drainage to inform on the presence of whirling disease. The ranch was also interested in the presence of whirling disease within this Ute Creek and Sangre de Cristo Creek system to inform on future locations for reclamation. We collected fish heads from 15 sites across the ranch. At each site, we collected 10 to 30 fish, which mostly consisted of brook trout. Because whirling disease manifests in the cartilage of fish, we focused our sampling efforts on smaller young fish. From this fish, we collected their heads, which is where whirling disease spores can be found. To quantify the spores, we processed heads using pepsin trypsin digestion methods, or PTD. This is a 3-step process that involves warming these heads in a water bath so they become more malleable. And then we smush them to oblivion and collect the cartilage and bones within the head. We then digest the bones in various chemical baths, which reduces them to a liquid. And from that liquid, we can then obtain a whirling disease spore count. To count the spores, we take the bone liquid and add a small amount of it to a grid like this one. We then count the spores within the grid, and using this equation, we could extrapolate the number of spores per grid to the number of spores per fish. So here's a breakdown of the infection prevalence per watershed. And before we dive into this, I just want to mention that all of the fish we collected had no clinical signs of whirling disease. So none of them looked abnormal, having black tails or swimming in circles. To the naked eye, all of these fish looked healthy, but there's obviously infection within these watersheds. Within the Trinchera watershed, we sampled 3 sites collecting 71 fish. The only site that came back positive for whirling disease was this lowermost site below all the barriers, which means that the reclamation locations had no signs of whirling disease. So stream segments above these barriers can be populated with native cutthroat once the non-native brookies are removed. Within the Sangre de Cristo drainage, we collected 49 fish, and these sites were hot for whirling disease, with a 67% infection prevalence. Because this infection severity at these sites, if any reclamation were to take place, the system would have to remain fishless in order to break that whirling disease cycle. But as of now, I don't think Trinchera has any plans to do anything with this drainage. And then finally, within the U Creek system, we sampled 9 sites and 4 came back whirling disease positive, for an 18% infection prevalence. I want to highlight this area of Ute Creek. These upper sites had no whirling disease detected, which is an interesting pattern based on the extent of whirling disease throughout the rest of the system. This could indicate that there's some sort of barrier preventing the spread of whirling disease, or maybe there are no worm hosts within these areas, or maybe whirling disease has just yet to spread up this far in the system. Whatever the reason, it's an interesting place to keep in mind for future management. In addition to fish head collection, we also collected water samples to test for environmental DNA of whirling disease. We collected eDNA at 20 sites, and most of these samples were taken at the same locations of fish head collection. These additional samples were collected from current cutthroat trout populations, places where CPW had already sampled for fish. So focusing on this big circle, this is the Placer Creek system where reclamation had already taken place, and has current populations of Rio Grande Cutthroat trout. Because of this reclamation, CPW keeps a close eye on this system. So we have a pretty good idea of negative and positive whirling disease locations. So above these barriers, we collected eDNA samples as known negatives for whirling disease. With this lowermost site below all the barriers as a known positive, and we know this because of CPW sampling efforts. So at each of our sites, we collected 3 1-liter samples of water. Each liter was then processed onto a filter, and these filters are what collect and house the DNA. The filters are then digested into a liquid, and we can extract DNA and amplify it to get a reading of how much DNA is found per liter of water. Through this DNA amplification process, we can create these curves. So when DNA is detected, these curves begin to form, and the earlier a curve forms, the more DNA is present within the system. These purple curves are known amounts of DNA. We know the amount of DNA in these samples because they are a standard control. You can literally just buy these known amounts of DNA off the internet. These green curves are our samples, so what we collected from Trinchera Ranch. By having this known amount of DNA and comparing it to our samples, we can estimate the amount of DNA within our samples. Oh, this dotted line is the threshold. So when curves go beyond this line, we consider them positive. You can see how our DNA sample is amplified later in the process, indicating less DNA. However, they did go above the threshold curve, so we do count those as positive samples. So for each of our 20 sites, we created 18 curves per site, for a total of 360 curves. Of those 360 curves, only 26 made it past the threshold, representing 5 sites that were positive for whirling disease. Of those 5 sites, I have how many times each of those curves amplified. And as you can see, none of them went 100% for detecting DNA, meaning that some of these curve amplifications, DNA was not detected, even at these sites that we knew were positive for fish. This is a little concerning regarding the effectiveness of eDNA testing. You would think that sites that came back positive for fish would also come back positive for DNA. However, that was not the case for our sampling efforts. As a reference, we had 8 fish head sites that were positive for whirling disease, with these Sangre de Cristo sites being especially hot. And of these sites, our DNA results only had 1 site that came back whirling disease positive, even though our fish heads picked up hot samples at all of these sites. Originally, we wanted to see if we could compare spore counts from fish heads to eDNA sampled within the water. However, eDNA sampling wasn't mimicking what we collected with fish heads. Of the 8 fish head sites, eDNA was only detected at 4 locations, in addition to that 1 Placer Creek site that we knew would be positive. And of those positive sites, eDNA detection was really low, indicated by those curves peaking later in the amplification process. So what does this mean for management? Well, through our sampling efforts, we found that searching for and testing the worm host was a difficult way to detect for whirling disease. Fish head collection proved to be the most effective way for testing for whirling disease, where we were able to obtain a spore count and gain an infection prevalence per watershed. However, these methods are lethal, which may not be ideal for sampling locations inhabited by cutthroat trout. eDNA detection did work, however, it wasn't super reliable with replicating what we found with fish head collection. So it can be an alternative to lethal collection methods, but management decisions shouldn't solely rely on eDNA detection. It's a great first step for detection, but additional methods need to follow in order to be certain about the presence or absence of whirling disease. eDNA is still a relatively new method for detection, and because of its new development, there are still a lot of uncertainties associated with it. But there are also a lot of advancements developing with this method. So what does this mean for Trinchera Ranch? These current populations of Rio Grandes have a promising future, especially the resident groups found above natural waterfall barriers, or within these reclaimed areas like Placer Creek. These reclamation locations have tested negative for whirling disease, meaning Rio Grandes can be reintroduced to these sites once the threat of non-native fish is removed. Knowing the spread of whirling disease throughout these systems can inform us on future reclamation or inform us on where to do more in-depth sampling. This upper section of Ute Creek could be the new focal point for reclamation because of the absence of whirling disease. Reclaiming this area would provide more habitat for future populations of Rio Grande Cutthroat trout. All of these efforts are to ensure the longevity and continued success of the Rio Grandes, and Trinchera Ranch is a promising location for their future because of the expansiveness of the property and their willingness to contribute to their conservation. And all of this couldn't be possible without all of the wonderful people that helped me through this. People at the testing lab at CSU, the wonderful people at Trinchera and CPW, my family and friends, and of course, all of you. Thank you for your support and your belief in my research, and thank you for choosing me as a recipient of the Steve Bailey Memorial Scholarship. And with that, I'll take any questions. Um, it's so what was the timeline of whirling disease? It's temperature dependent. So depending on, I don't know the exact temperature that it like peaks at, but depending on the temperature of your streams, it does become more prevalent in the system. But definitely when it's warmer. So I would say like summer, like August, I would say is like peak infection season. Yeah, so the question was how long do the trout have to be out of the system for whirling disease to be eliminated? And from that Placer Creek system, I think it was a year and a half before they started. So they placed fish in sentinel cages, which I don't know if you guys know what sentinel cages. It's just basically like an in-stream aquarium, and then they just subject these fish to the stream. And if they do get infected, then whirling disease is still there. If not, then they considered it eliminated. And I think it was about a year and a half for that Placer Creek system. This will be my third and final year with this project. Yeah.
It's always thought that the eDNA technology substitutes into sacrifice fish. Doesn't sound like that's the case. If you take fish heads and just measure, say, you switch them out and you measure eDNA. That's okay. The question is it the technology and sensitivity of measuring eDNA, or is it sampling issue, or what do you think the disparity is?
Yeah, so the question is how come eDNA isn't as effective as we thought? And I think part of it is a sampling issue. So the standard way to collect DNA from water is just collecting a grab sample from within the water column. But based on the life cycle of worm disease, we know that these spores and these worms inhabit like the benthos of the stream. So I don't know if it would make a difference if we were to accept or sample closer to the bottom of the stream, and then we would get higher concentrations of DNA. But I do really think it is a sampling error, or just like we don't know how to do it quite yet. Uh, yeah, so they do the same test I did for my water samples of DNA. They do also do with fish heads themselves. It's a qPCR if anybody knows what that is. But yes, they do show up a lot more positive than just grab samples of water. Yeah, I think it's just based on like there's just low concentrations, at least in my samples, there's low concentrations of whirling disease DNA in the water versus if we were to DNA test the fish heads, the positive ones would obviously have peaks in them. Yeah. Yes, but in the back. What is what is Chinchera Ranch? It's just a huge private ranch, uh, located on Lovita Pass. If any of you guys know where Lovita Pass is, um, they're part of like the Sangre de Cristo. I don't know if they're part of it, but they're at least associated with the Sangre de Cristo land grant, and it's just like this huge area through the Sangre de Cristo mountains that is either public, so there's national park there, there's national forest, there's Trinchera Ranch, and then there's more national forest. And all of that area is like a conservation area, and the ranch is just really involved and excited about conservation. Yes. I would love to work for the state. Like, I would love to work for CPW. Um, However, it's hard. Nobody wants to retire from those jobs. So it's hard to get in there, but that's my goal is to work for the state. Yes. Yeah, so that's a really good question. The question was, can we apply what we found for bugs throughout the state? And it depends, I guess. It always depends. Based on the environmental variables that we collected, it could always differ if somebody else collected more or less than we did. Um, I'm actually curious to see how that does relate because just on the other side of the mountains, in the Great Sand Dunes, they did a similar bug, which is actually what I based all of my bug sampling methods on. They did a similar bug study, so I would like to see the comparison, that how my bugs to their bugs is. But in terms of throughout the state, I don't think I would trust it to be applicable, but it depends. Yeah, but that was a brown trout. Yeah. I would love to say that was caught on a fly, but we actually we shocked that fish. Yeah. That's the only way you could catch the big ones. That was when I worked on the Blue Valley Ranch in Kremling. So I don't know if any of you float the blue, but that's where that fish came from. Yes. Any more questions? Yeah. Uh, yes and no. So yes, there is a lot of stats involved with my stuff, especially my bug stuff. There's a lot of stats involved. My advisor is really good at what he does, and he was able to help me figure out all of the math associated with my bug stuff. In terms of the DNA stuff, um, that's just computer does it for us. The computer makes those curves and tells us a number. So the hard part was collecting and digesting the fish heads. So each summer I spent about a month and a half on the ranch collecting fish heads and bugs and eDNA samples. Any more questions? All right, thank you guys so much for having me.